Hi everyone, and welcome to our video about chloroplasts and photosynthetic pigments. So this is going to be the second video in our photosynthesis topic, and what we're going to be looking at here are two spec statements: 5.2.1b, the structure of the chloroplast and the sites of the two main stages of photosynthesis, and then 5.2.1c, first part of it, the importance of photosynthetic pigments in photosynthesis. The practical, which comes up in part 2, we'll have a look at as a separate entity. To start off with then, let's have a little look at chloroplasts. Now, chloroplasts are something that we've heard about since year 7, but when we were talking year 7 knowledge, we literally said they're green because they contain chlorophyll, and there ended our knowledge. Sadly, at A-level, we can't get away with just that. We do need to add a little bit more detail to our knowledge in order to be able to succeed in the A-level papers. So, first of all, they're still called chloroplasts. That's a nice thing. And these are disc-shaped, approximately 2 to 10 micrometers in length. Now, what we find when we look at the actual structure of the chloroplast itself, it doesn't just have a single membrane, but it has a double membrane. Now, that's what we refer to as the envelope. What we then have between the inner and outer membranes is basically a little space, which we call the intermembrane space. It's quite logical. So inter basically means the between, and then membrane clearly means membranes. And it's a space between them. So quite simply, inner and outer membranes and a little space between them called the intermembrane space. One thing we should know about a difference, if you like, between the inner and outer membranes is that the outer membrane is highly permeable. And this is going to be important as we start to understand how chloroplasts actually work. If we now go beyond those inner and outer membranes, we're now going to delve inside the chloroplast a little bit more, then we find two new phrases that we're going to have to get our heads around. The first thing is this stuff called the stroma. Now, that is a fluid-filled matrix. We'll look at this in more detail in a moment. And then we have these things called grana, which are stacks of thylakoid membranes. So if we have a little look at the picture down here, what we can see, we've sliced it open. So we have our outer membrane first of all, which is this outer part here. We then move in, and this is our inner membrane. So you can see that double membrane there, the envelope. When we come inside, we then get to our stroma, which is that fluid-filled matrix. And then you can see there's these things that look kind of like little stacks of coins in the middle there. Now, those are the grana, okay? So we have singular granum, plural grana. And they're made up of these individual little discs, which we will refer to as the thylakoids. Now, the only reason that we know that this is what it looks like inside a chloroplast is because we have electron microscopes. This would not have been possible in the past pre-electron microscopes. We just would not have been able to get the magnification necessary to see that level of detail. If we now consider the grana themselves, so we can see in the bottom corner here I've given you a simple little diagram of a granum, then connected to something else, another one basically. Now, what we have is the grana are the site of the light-dependent stage of photosynthesis. Back at GCSE, you should have been told that photosynthesis is a two-stage process, light-dependent and light-independent. So the light-dependent stage, the bit that needs the sunlight, this happens in the grana. Now, our thylakoids that make up the grana are these flattened disc-like sacs. Okay, so that's pretty much how we describe it. And what we then do is we stack up all of those little thylakoids, all those little flattened disc-like sacs, one on top of the other, to make an individual granum. Many of them grana. Now, these are not just little individual stacks just sitting around in the chloroplast on their own. Instead, these little granum can actually be connected to a second granum through this structure called an intergranal lamellae. So inter is between, granal referring to our grana, and lamellae is just the name we're going to use there. And we can see it in the little picture in the bottom left there. So we've got two of our little stacks of thylakoids, so two grana, and then if you look between them, there's this little region which is the intergranal lamellae. Okay, so that is showing this little interconnection between those individual stacks. I would hope from the little diagram that we've seen, it goes without saying that we don't just have a single granum in any chloroplast, we have many grana, and each cell has many chloroplasts. So if you imagine there's many chloroplasts with many grana within each of them, we then have this really quite significantly large surface area for the distribution of all of these key bits needed for that light dependent reaction. And the key bit we're going to focus on is this thing called photosystems. Now we can obviously understand a little bit about that because photo is obviously just telling us it's all to do with light, and a system is something that's working together. So basically, by having this really large surface area caused by the many chloroplasts with the many grana, then we've got plenty of space to have these photosystems in our actual plant cells there, and other cells that contain chloroplasts, that will therefore allow us to trap the light energy and then allow us to generate our ATP through the use of this thing called ATP synthase. And obviously because it ends in A's, we know that it's an enzyme. Now, if we just think about the last thing, we've not really mentioned in detail there, the stroma, then what we have within our chloroplast, we've got all our little grana, and then the stroma basically surrounds the grana themselves. Now, the reason for that is it's going to make it really easy then for the products of photosynthesis to then cross into the stroma, which is the location of that second stage of photosynthesis, the light independent stage. So what we find, we've got these two different regions for the two stages of photosynthesis within the chloroplast, and we've got it set up so that we can get that easy transfer of the products from one stage to allow the second stage to take place. We will look at the actual process of photosynthesis in more detail in a future video. Now, the stroma, as we said at the beginning, is a fluid-filled matrix. Now, what that means is that we've got this substance that's going to contain all of the enzymes that we need for that light independent stage. We are going to have DNA present in there. We will find starch grains present in there, and we're going to have small ribosomes. And these ribosomes are going to be really important because they're going to then assemble the proteins that are needed for that process of photosynthesis. And of course, hopefully we remember from our biomolecules unit that enzymes are of course proteins. Before we move on to the photosynthetic pigments, it is worth mentioning the origins of the chloroplast, because this comes up every once in a while. And what we believe to be true is that chloroplasts used to be free living prokaryotic cells. So they used to exist as their own little prokaryotic entity. Now, at some stage in the distant past, they ended up incorporated into a eukaryotic cell, and now they've got this endosymbiotic relationship. So again, break that word down. Endo obviously means inside. Symbiotic is all to do with basically working together. So what we actually have here is this like codependent, I suppose is a way we could view it, relationship where we've got these two parts that are now working together, but they've become one with one another. So our little original free living prokaryote has now become part of that eukaryotic cell. You will of course want to know what evidence there is for this. Am I just making this up just because it sounds quite cool? Well, no, we do have evidence. So first of all, the chloroplasts are produced by division of other chloroplasts only. So the only way we can make more chloroplasts is from others. You can't generate a chloroplast from just the eukaryotic cell without the chloroplasts. They've got their own genome, which has a circular loop of DNA. Think about where we've heard of a circular loop of DNA before. Prokaryotic cells, when we looked at those, remember. They've got their own 70S ribosomes and their own mechanism for protein synthesis. So they've got these 70S ribosomes, same as in the prokaryotic cells, remember, and that means they can actually carry out protein synthesis on their own. They've got this whole mechanism for doing it without any other parts of that eukaryotic cell. And when we look at the pigments that are found within our chloroplasts, they're really similar to those that we find in the cyanobacteria, so another type of prokaryote. So what we've got there are 4 key bits of evidence that support this idea that our chloroplasts were once these free-living prokaryotic cells. The last bit that we're going to look at in this video are the photosynthetic pigments. Way back at GCSE and before, we've always described our chloroplasts as containing chlorophyll. This is one of those moments where, yeah, we lied. It wasn't that simple after all. Good news, there is still chlorophyll in there, just a bit more complicated than we led you to believe lower down the school. Sorry. Now, before we delve into the exact pigments themselves, what we're going to do is actually have a look at these things called photosystems. So we've mentioned those where we were looking at the structure of the chloroplast. We said that the photosystems are within those thylakoid membranes. Remember, we talked about the large surface area. This is here. Now, when we talk about a photosystem, this is basically what we're talking about in that bottom corner. So we've got a funnel shaped structure, and in that funnel shaped structure we've got photosynthetic pigments. So pigments that are all to do with using light to create something. We have different pigments and they will absorb different wavelengths of light, and what we there find is that that energy that is being absorbed from the light initially is then going to basically be filtered down through each of those different little parts until we get to the bottom there. And at the bottom we have this primary pigment reaction center. Now, chlorophyll, the generic term we have used all the way down in year 7, 8, etc., is actually a mixture of pigments. Now, what we do know is that they all contain this porphyrin group, and that contains an atom of magnesium. In addition to that, we've also got this long hydrocarbon chain that makes it up. Now, this is one of those bits that we've looked at previously where we said that if you have a lack of magnesium, then it's not going to produce the chloroplasts, probably was how it was phrased lower down. The reality is that that lack of magnesium for our plants means that there's going to be a limit to the amount of chlorophyll that could actually be synthesized. And that therefore means the rate of photosynthesis will be limited. Because if you've limited the chlorophyll, you've limited the photosynthesis because you've limited the amount of light that can then be harvested. What we're going to do then is delve into the exact pigments in a bit more detail now. First up is chlorophyll A. Now, chlorophyll A is actually occurring in 2 forms. So they're both chlorophyll A. They are both blue-green in their appearance, and they're located at the center of the photosystems. Because they're blue-green, they're going to absorb red light, so light from the red end of the spectrum. What we find then is because we've got these 2 forms of chlorophyll A, then we are going to find P680 in photosystem 2. Now, the reason we call it P680 is because this has a peak absorption at the wavelength of 680 nanometers, hence 680. Then in photosystem 1, we find P700, which, logic dictates, has the peak absorption of 700 nanometers there. So basically what we've got here are 2 different forms of chlorophyll A both absorbing light from the red end of the spectrum, but with different peak absorptions, one at 680 nanometers, the other at 700 nanometers. And therefore what we actually find is we've got these two photosystems, very creatively named photosystem 1 and photosystem 2, with those different types of chlorophyll A at their centre. It is worth mentioning here that it's not just red light, there is some blue light of 440 nanometers which can also be absorbed by this chlorophyll A. The next pigment we need to know about is chlorophyll B. Now chlorophyll B actually has two absorption peaks. You'll find one between 400 and 500 nanometers and the other at around 640 nanometers. When we look at chlorophyll B, this is the one that appears yellow-green in colour. So what we can see, we've got clear differences between chlorophyll A and chlorophyll B. They're different colours, they absorb different wavelengths of light, and therefore they're both going to be useful to the plant because with the two of them, they can absorb a wider range of wavelengths of light from the sun and therefore provide more energy for that process of photosynthesis. We don't just have chlorophyll A and chlorophyll B, we also have some other pigments present called accessory pigments. Now there are two that you should really be aware of here. We've got the carotenoids, which absorb blue light 400 to 500 nanometers, and then the xanthophylls, which are going to absorb the blue and green light, which is the 375 to 550 nanometers. So what we actually find here then is that with the combination of chlorophyll A, chlorophyll B, our carotenoids and the xanthophylls, we can absorb quite a large number of wavelengths of light. So this means that we're able to extract, if you like, a maximum amount of energy from the sun that hits that leaf. So what we've got here is just a graph showing us what three of our little photosynthetic pigments are capable of absorbing in terms of the wavelengths of light. So we've got chlorophyll A, chlorophyll B and carotenoid on there. On the x-axis we have the wavelength of light, down 400 on the left hand side there, up to 700 nanometers on the right, so going from blue to red in the spectrum, and the y-axis is just giving us the amount of light that is absorbed by each of those pigments. If we look at chlorophyll A first of all, what we said was that it's absorbing some blue light around about 440, and if we have a look at this line here, we can see we get a peak round about 440. We then said that we've got those peaks around the 680 or 700, and again, here we go. There's our chlorophyll A second peak. We then said our chlorophyll B is going to absorb around the 400 to 500 nanometers, so there is our peak at that end, and again at the other end we've got a peak around about 640 nanometers. So supporting what we've said. If we then have a look at our carotenoid, we can see in this case we have a peak down here, probably sitting around on a glance about 480-ish I would guess, but we have our little peak between our 400 and 500 nanometers again. So what we can see is that between just those three pigments, we've got 5 peaks, so 5 different wavelengths of light that can be absorbed, which means that our plant is therefore gaining a significant amount of energy that can then be utilised in photosynthesis. As always, I do suggest that you subscribe to the channel so you can see when I next upload a video, and head on over to the A-Level Biology website to see what other resources I've put up to help you with your A-Level Biology studies.